First of all, on behalf of our team, which include Sachiko Sato, Jerome Franet, and myself, I'd like to extend our heartfelt thanks for your financial support. Your backing has been instrumental in our endeavor to develop a saccharide-based therapy using N-Acetylglucosamine to mitigate Duchenne muscular dystrophy. I would like to discuss briefly the science that underpins our research. Oligosaccharides, which are small chain of the sugar on the muscle membrane, play an essential role in maintaining a healthy muscle physiology. This ranges from muscle regeneration to the muscle construction. The muscle surface dystroglycan complex connected to dystrophin inside the cells carries more than 50 unique oligosaccharides. These oligosaccharides are crucial as they bind to a cell matrix protein called laminin, an interaction fundamental for healthy muscle functioning. Interestingly, while each sugar chain binding to laminin is rather weak, they form a robust and flexible bond. This synergy creates a stable yet adaptable connection within muscle tissues, illustrating the power of unity in the biological structure. Now, let us visualize the concept we just discussed using a close-up of the vector. In this video, you witness an example of how a collection of weaker elements can create remarkable robustness and flexibility. I invite you to imagine a similar action occurring at the microscopic level in our muscles. This is akin to the interaction between a sugar chain of the dystroglycans and the laminin. Just as demonstrated in this video, collective effects of the many weak bonds can lead to a strong and adaptable system. This principle is crucial in understanding how we aim to harness the power of the oligosaccharides in treating the DMD. Now let's examine the key issue in Duchenne muscular dystrophy. In Duchenne muscular dystrophy muscle, the essential saccharides we've been discussing are notably missing. This is due to the absence of the entire dystrophin-dystroglycan complex in the Duchenne muscular dystrophy muscles. Without this complex, there is a significant disruption in the muscle function and regeneration. This lack of functional saccharide is one of the major factors contributing to the symptom of the DMD. Having understood the absence of specific oligosaccharide in DMD muscles, let's explore an alternative approach. Apart from the oligosaccharides associated with dystroglycans, muscle membranes are also decorated with many other types of oligosaccharides presented by the membrane proteins of the muscles. In addition, the cell matrix protein laminin itself carries more than 38 different oligosaccharides. This raises us an intriguing question. Could we possibly use these other oligosaccharides as a substrate for our ones missing in DMD? More specifically, can we enhance the biosynthesis of these alternative oligosaccharides and utilize other host proteins to cross-link them? We decided to focus on galactin-3, a member of the galactin family which is known to be abundantly expressed in the muscle tissues. Galactin-3 has a potential to bound and to cross-link those oligosaccharides of the laminin and oligosaccharides of the muscle membranes, providing these oligosaccharides are optimal for galactin-3 binding. Thus, we explore the possibility of using a saccharide called N-Acetylglucosamine, or gluconac for short, to increase the biosynthesis of the oligosaccharide for galactin-3 binding. Let us now take a closer look at the biochemical pathway of how the gluconac is uptaken and functions within our cells. Gluconac is rapidly taken up by the cells and play a crucial role in producing oligosaccharides that are optimal for galactin-3 binding. It's important to note the distinct pathway of the N-Acetylglucosamine compared to its relatives, glucose and glucosamine. While glucose and glucosamine are absorbed through glucose transporter and primarily utilized either for energy generation or for glucose storage, glucagon operates differently. Glucagon is directly incorporated into the oligosaccharides. This means the glucagon bypass the most common pathway of energy production and storage, directly contribute to the formation of the crucial oligosaccharide synthesis that is important for glycine 3 binding. Based on the unique property and pathway of the glucagon we just discussed, we hypothesize that utilizing the 100% pure N-acetylglucosamine as a treatment could significantly impact the muscle health in those with a Duchenne muscular dystrophy. We believe that this approach can potentially foster an optimal muscle oligosaccharide environment. This environment is crucial for dynamic muscle activity, which is often compromised in DMD. Additionally, we anticipate that pure N-acetylglucosamine could enhance muscle regeneration process. This aspect is particularly important given the rapid muscle regeneration observed in DMD. Now, let's move on and where our research stands at the present. Previously, we demonstrated that intraperitoneal administration of the glucagon can reduce the disease progression in a DMD mouse model. Building on that, we have now developed a live cell single-cell tracking methodology. This advanced technology allows us to closely examine the process of myogenesis, which is a muscle regeneration, using the primary muscle cells called myoblasts. Utilizing this single-cell tracking technology, we found that glucagon treatment enhances the myogenesis. This is evident through the promotion of the initial movement of the muscle cells and the fusion of the muscle cells, key process in muscle repair. Let us now look at our preliminary finding from in vivo preclinical study using a DMD mouse model. Our first exciting outcome was observed with 1 month oral administration of N-acetylglucosamine. This treatment led to reduced muscle damage and enhanced daily movement in the mouse. Notably, we've identified 2 optimal doses for this effect. We completed a 1 month treatment combining the N-acetylglucosamine with prednisone. The analysis of the muscle tissue from this study is currently underway, and we are very eager to see the results. Finally, in a long-term study, a 14-month oral glucagon treatment concluded without showing any significant toxicity or adverse effect. Similarly, the mortality rate between the control and N-acetylglucosamine treated group were comparable. We are also in the process of analyzing the muscle tissue from this extended study. I'd like to take a moment to specifically highlight the substrate at the heart of our research, N-acetylglucosamine or glucagon. We are examining the effect of 100% pure N-acetylglucosamine on DMD muscle tissues and the myogenesis process. This is a precise and targeted study. It is important to note that N-acetylglucosamine is chemically distinct from glucosamine or glucosamine sulfate which are commonly available over the counter in pharmacies. Our preliminary observation indicates a notable difference in efficacy and safety. Unlike glucosamine, which does not promote myogenesis and may be even toxic, glucagon shows promising results without these drawbacks. Furthermore, while the glucose and glucosamine can lead to detrimental protein modifications called glycanation, a process potentially harmful to the cells and muscles, glucagon does not trigger these harmful reactions. This makes it safer and more viable option for our therapeutic goals. To conclude the presentation, may I recapitulate the key findings and implications of our research. Our preliminary results suggest that oral administration of N-Acetylglucosamine or glucagon is effective in mitigating the progression of DMD. It is important to emphasize that positive effect we observe especially with 100% pure N-Acetylglucosamine. Once proven in the further studies, this treatment could potentially be applicable to all DMD patients, regardless of their specific genetic mutations. Moreover, N-Acetylglucosamine-based therapy holds a promise for combination with other therapies, both non-targeted and targeted, because its molecular mechanism is likely distinct from most of these therapies. Thank you for your attention. In summary, our preliminary results suggest that oral N-Acetylglucosamine treatment is effective. Please note that effect was confirmed only by the 100% pure N-Acetylglucosamine, but not the one found in the supplement quota of a pharmacy. Once proven, this treatment could be taken by all Duchenne muscular dystrophy patients, regardless of their mutations. In addition, we expect that N-Acetylglucosamine-based treatment could be combined with other non-targeted and targeted therapies, as the molecular mechanism is likely very different from most of those treatments. Thank you.